So what about sex? So it turns out that you can do experiments where people have orgasms while they're in the brain scan. And these experiments have mostly been done in the Netherlands. That's true. I didn't make that up. And I know, I can just tell by looking at you that you have a very good, imagination. So I want you to use your power of imagination to imagine just how deeply unsexy this experiment really is. So you're in the brain scanner. You're lying back. The brain scanner is this big, clanging, horrible metal donut, and your head is stuck into it. And because if your head moves, it ruins the image, your head is strapped down with this big, heavy Velcro strap onto the board. All right? And then your nether regions are sticking out into the room where there's a bunch of geeks in white coats who look like me, writing things on a clipboard. And, there's an IV in your arm. Because in order to do this kind of imaging, you need to inject a bolus of radioactive water. And, there's no really polite way of saying this. There's a rectal probe involved. And I don't know about you, but I'm a big fan of the alien abduction story, so even saying the words rectal probe makes me a little happy. So why is the rectal probe involved? Well, the reason is because, we want to make sure the females aren't faking it in these experiments. And so in order to know that, it turns out that in both men and women, during orgasm, there is a very particular 10 hertz contraction of the rectum, and it can't be faked. So in a separate experiment, they have women experiencing orgasm. You record this flutter in the tension of the muscles in the rectum, and then in a brilliant move, they have women try to do their best fake orgasm, Harry Met Sally sort of situation. And the flutter isn't there. So, all right, thinking you push, you're lying back there, and then your sweetheart is out there trying to bring you to orgasm. Now, initially, guys couldn't do these experiments. They would just wait. And it wasn't until the advent of the phosphodiesterase type 5 inhibitors, the erection drugs, that guys really were able to participate. So the first experiments were done with women. And so, there's your sweetheart out there trying to stimulate you to orgasm. And then when you're getting close, you say, okay, I'm close. Inject the radioactive water in my arm. And the technician says, okay, I'm injecting the radioactive water in your arm. You now have a minute to have your orgasm. Just relax and let it happen. And so it's kind of astonishing that anyone can achieve orgasm under these conditions. But they can. And it turns out that actually orgasm in men and women from a brain scanning point of view is remarkably similar. It's much more similar than dissimilar. And not surprisingly, you see a strong activation of the medial forebrain plexus circuit. You also see in the brain there is a map of the body surface. So if we touch your elbow, you can see the elbow map light up. And not surprisingly, the genitals are lighting up. Now there are also some parts of the brain that are being deactivated at the moment of orgasm. So a region called the parieto-temporal junction that is involved in social evaluation and cognition, very deactivated. Your medial prefrontal cortex, that is involved in executive function, moral choice, and decision making, very deactivated. So I guess if you're looking for use, you can use, you can use. I can't even speak. Thank you so much. If you're looking for news that you can use, I would say that the moment of orgasm is not the point where you should be allocating the stocks to your retirement. Because there's a lot of deactivation. So we've got this evolutionarily ancient circuit. It evolved so that we would eat food, drink water, and have sex and survive and pass our genes on. But clever us and clever other critters, we have figured out how to activate it artificially. So I'm activating it artificially right now with a little bit of alcohol. So, nicotine, cannabis, alcohol, cocaine, Heroin, amphetamines, all of these drugs activate the medial forebrain pleasure circuit. Some of them, like cocaine and amphetamines, do it in a very direct way. Other ones do it in an indirect way. But you might be thinking, oh, well, that's a big long list of drugs. I get it. All psychoactive drugs activate the brain pleasure circuit. Well, that doesn't not to be true, actually. So, for example, hallucinogens like LSD and mescaline don't activate the brain pleasure circuitry. And antidepressants of the SSRI family, like Prozac and similar drugs, don't activate the brain pleasure circuitry. And so, actually, there's a perfect correlation. If a drug doesn't activate dopamine release within this pleasure circuitry, it has no potential for addiction. That's why LSD isn't addictive, for example. Whereas if it does, then it has a varying risk. And we'll talk about that more in detail as we consider addiction later on. So we didn't evolve to, take some tobacco plants and set it on fire in our mouth and inhale the fumes, or drink alcohol or take any of these other drugs. We've just been clever in figuring out how to short circuit, how to artificially activate this evolutionarily ancient pleasure circuit in these ways. And interestingly, it's not solely a human compulsion. And so the second section that I'd like to read off the page, the last section I'd like to read off the page, is about this. Lord Byron, the British romantic and satiric poet of the early 19th century, wrote, "Man being reasonable must get drunk. The best of life is but intoxication." However, intoxication with psychoactive drugs is not an exclusively human proclivity. Animals in the wild will also voluntarily and repeatedly consume psychoactive plants and fungi. For example, birds, elephants, and monkeys have all been reported to enthusiastically seek out fruits and berries that have fallen to the ground and undergone natural fermentation to produce alcohol. In Gabon, which lies in the western equatorial region of Africa, boars, elephants, porcupines, and gorillas have all been reported to consume the intoxicating hallucinogenic iboga plant. There's even some evidence that young elephants learn to eat iboga from observing the actions of their elders in the social group. In the highlands of Ethiopia, goats cut the middleman out of the Starbucks business model by munching wild coffee berries and caffeine buds. But we have to be careful here. Do we really know whether these animals like the psychoactive effect of the drug, or are they just willing to put up with them as a side effect of consuming a valuable food source? After all, fermented fruit is a tasty and nutritious meal. While it's hard to dissociate these motivations in animals, many cases suggest that the psychoactive effect is the primary motivator for consumption. Often only a tiny amount of the plant or fungus is consumed, so while its nutritional effect is small, its psychoactive effect is huge. Perhaps the most dramatic example of non-nutritive animal intoxication is found among domesticated reindeer. The Chukchi people of Siberia, who are reindeer herders, consume the bright red hallucinogenic mushroom Amanita muscaria as a ritual sacrament, and their reindeer also indulge. Having discovered the mushrooms growing wild under the birch trees, they gobble them up and then stagger around in a disoriented state, twitching their heads repeatedly as they wander off from the rest of the herd for hours at a time. The active ingredient of the Amanita mushroom is called ibotenic acid, a portion of which is converted into the body into another compound called mucimol, the substance that actually produces the hallucinations. What's interesting about ibotenic acid is that only a fraction is metabolized in the body to form mucimol, while the rest, about 80% of that consumed, is passed in the urine. I think you can see where we're going, and it's very disgusting. Reindeer have learned that licking ibotenic acid-laden urine will produce as much of a high as eating the mushroom itself. In fact, this drug urine will attract reindeer from barn-wide, and they will even fight over access to a particularly attractive patch of yellow snow. All of this has not gone unnoticed by the Chukchi, who collect the urine of their Amanita-eating shamans for two purposes. The first is simple thriftiness. Amanita mushrooms are often scarce, so urine recycling can provide about 5 doses for the cost of one fresh one, albeit with a rather severe aesthetic penalty. The second is that the reindeer are just as enthusiastic about human Amanita-tainted urine as their own, And so they can be effectively rounded up with a bit of stuff sprinkled in the corral. Clearly, Siberian reindeer are not fighting over drugged urine for its nutritive